Hello and welcome, everyone, for this presentation on the ICH Q5A R2 guideline, viral safety evaluation of biotechnology products. This session will cover the key updates, regulatory expectations, and practical case studies that professionals in biotechnology, pharma, and regulatory affairs need to understand. By the end of this session, you should have clarity not only on the scientific principles, but also on how to apply them in real-world manufacturing and submissions. Let's begin with the background. The original Q5A guideline was finalized in 1999. Since then, biotech has moved forward drastically. We now have gene therapies, viral vectors, and nanoparticle-based vaccines that weren't part of the earlier scope. At the same time, new tools like next-generation sequencing, or NGS, have transformed virus detection. This revision, Q5A R2, was adopted in November 2023. It reflects the evolution in manufacturing, analytical science, and a need for global harmonization. The ultimate goal is ensuring patient safety while enabling efficient product development and approval. The objectives of Q5A R2 are simple but powerful. It harmonizes viral safety evaluation globally and focuses on three complementary approaches: testing and qualifying cell lines and raw materials, assessing the virus clearance capacity of manufacturing processes, testing products at appropriate stages of production. Together, these three layers provide a strong safety net against viral contamination. So what has changed? First, new product types like AAV gene therapy vectors, baculovirus-derived products, and virus-like particles are now explicitly in scope. Second, continuous manufacturing gets its own section for the first time, recognizing its unique risks. Third, molecular methods, particularly NGS, are formally introduced as acceptable, even preferred, alternatives to animal-based testing. We also see flexibility with resin reuse and the ability to leverage prior knowledge across platforms. All of this makes the guideline both stricter on viral safety and more pragmatic for industry. Let's look at how this applies in practice. Case Study 1. AAV vectors produced in HEK 293 cells via plasmid transfection. Case Study 2. Baculovirus-expressed AAV vectors in SF9 insect cells. Both involve new product classes with higher complexity. Q5A R2 guides us on assessing raw material risks, testing cell banks, selecting model viruses for clearance studies, and applying new methods like NGS. This demonstrates how the guideline is directly applicable to today's gene therapy and vaccine platforms. This case study focuses on monoclonal antibodies from CHO cells, a mature product type. Here, prior knowledge is key. For example, using historical data from the master cell bank reduces redundant testing. Low pH inactivation for retroviruses is a well-established clearance step, and resin reuse studies can be justified with existing data. The point is, Q5A R2 encourages us to avoid reinventing the wheel if robust, platform-based validation data already exist. Next, continuous manufacturing. Unlike batch production, CM involves longer cell culture durations and integrated unit operations. That creates new risks, like contamination persisting across multiple lots. The guideline emphasizes careful sampling strategies, virus clearance validation at each critical unit operation, and ensuring robustness under worst-case conditions. The takeaway is that viral safety must be designed into CM processes from the start, not bolted on later. Now, let's talk about next-generation sequencing. NGS can replace traditional in vivo assays and even antibody production tests. It is considered a limit test, meaning it detects the presence of viral genetic material broadly, without needing a head-to-head comparison with older methods. Benefits: broader detection, faster results, and alignment with the 3 rupees principle of reducing animal use. Challenges: validation requirements, and interpreting what a detected nucleic acid really means for infectious risk. But overall, this is the future direction. What does this mean for industry and regulators? Harmonization across ICH and non-ICH regions avoids conflicting requirements. Companies can adopt modern tools like NGS without fear of rejection. Costs go down and approvals go faster. At the same time, regulators get a clearer framework for evaluating viral safety across new modalities like gene therapies. It's a win-win for patients and stakeholders alike. As a conclusion, here are the practical takeaways. Start aligning your processes with Q5A, R2, early in development. Use platform validation and prior knowledge to reduce redundant studies. For continuous manufacturing, build viral safety into the process design itself. And finally, embrace molecular methods like NGS where possible. This is where regulators are clearly heading. Now, the comment box is always ready for the queries, and must not forget to get the reference documents for more detailed study from description box.